The overall goal of this synthetic biology protocol is to rapidly determine terminator functionality and quantify terminator strength in an in vitro system that utilizes Golden Gate assembly.
This protocol is used to test terminators that we found in silico using bioinformatics and test them in a biological setting. This allows for rapid and simple confirmation of bioinformatically identified terminators in a lab bench setting. The main advantage of this protocol is its speed. We can go from bioinformatic identification to biological confirmation in less than 1 week, and in doing so we can also verify that a terminator is in fact a terminator, and we can quantify its terminating strength.
Generally, individuals will initially struggle with this technique because it sounds counterintuitive to add the restriction enzyme with the ligase in the same tube. However, we use type 2S restriction endonucleases, and the recognition site for the enzyme is separate from the actual cut site. Once the terminator is ligated into the pGR-Blue Plasmid, this is a permanent ligation. However, by having the restriction enzyme and the ligase in the same tube, going through multiple cycles, we can achieve a high cloning efficiency.
Begin this procedure by resuspending the individual oligonucleotides in nuclease-free water at a concentration of 100 micromolar. Then, prepare 10x annealing buffer. Next, label a 1.5 milliliter microcentrifuge tube for each terminator to be tested. To each tube, add 16 microliters of ultra-pure water, 2 microliters of 10x annealing buffer, and 1 microliter each of the top and